Very good evening to all of you. Today we are going to do the challenge of biochemistry. Biochemistry has 16 MCQs, and if you are thorough in your practice regarding biochemistry, you will score you will mark all the 16 questions correct, and it will naturally push your rank to the top. Why I am telling you, because each subject has its own weightage and every subject is important to score a good rank. And this biochemistry, most of the students do neglect. They concentrate more on the clinical subjects like medicine, surgery, and neglect the biochemistry, anatomy, physiology, which takes it toll in the long run. So please focus on this subject also. So let's move to the first MCQ. Apo B48 is a remnant of. Apo B48 is a remnant of A. RNA editing, B. RNA interference, C. RNA splicing, D. DNA translocation. It is RNA editing. What happens in liver cell? ApoB100 gene gives rise to ApoB100 protein. But ApoB48 protein is made in intestinal cells from the gene encoding ApoB100 by the process of RNA editing, which is also known as differential RNA processing or chemical modification of RNA. This is a post-transcriptional modification. This RNA splicing is also a post-transcriptional modification, but this is removal of intron and not editing. RNA interference is a silencing technique where the gene is silenced by not letting the product of gene form. Translocation occurs during translation. This is the movement of ribosome over mRNA. So the correct answer here is A, RNA editing. Hepcidin decreases iron absorption by inhibiting A. Divalent metal transporter, B. Hep-histine, C. Ferroportin, D. Transferrin. What do you think is the correct answer? Take your time. What do you think is the correct answer? Now, what is hepcidin? This hepcidin regulates iron metabolism. It mainly regulates iron entry in the circulation. By inhibiting ferroportin, hepcidin reduces dietary iron absorption. The iron release from macrophages is also reduced by ferroportin inhibition. So increased hepcidin activity is responsible for reduced iron availability. Increased hepcidin will lead to anemia. Decreased hepcidin leads to iron overload. Hepcidin synthesis and secretion by liver is controlled by iron stores within the macrophages, inflammation, hypoxia, and erythropoiesis. Now, regarding the other options, divalent metal transporter. It absorbs divalent metal ions from intestine, especially ferrous. Hep-histine. Hep-histine is involved in homeostasis of iron and copper. Transferrin is a protein which is responsible for the transport of iron. So the correct answer here is A, C, ferroportin. Which of the following has the highest glycemic index? A, glucose, B, fructose, C, sucrose, D, sorbitol. Now what is glycemic index? Glycemic index is the increase in blood glucose after the test dose of a carbohydrate compared with that of an equivalent amount of glucose. This glycemic index is a value assigned to foods based on how slowly or how quickly those foods cause an increase in blood glucose level. The highest glycemic index is for glucose and galactose. So the correct answer here is A, glucose. GLUT5 is a transporter for A, galactose, B, fructose, C, mannose, D, glucose. It is fructose. GLUT5 is for the transport of fructose. Let's move on to the next question. Please click the link and go to the description area. In the description area, there is a link. Short link is there. Just click on the link. You will reach a WhatsApp group. This WhatsApp group belongs to Ultimate Neat PG Breakthrough Blueprint. This will this masterclass covers all the 19 subjects in just 2 days in a holistic manner. It will help you if you are a beginner. It will help you speed up your preparation. It will guide you how to prepare. And if you are at the fag end and you are giving the exam right now and want to secure a good rank, then also it will be very beneficial for you because it will speed up your revision process. So please click the link in the description box to join Ultimate Need PG Breakthrough Blueprint. The first 10 people who click the link in the description box will join the WhatsApp group. They will get this masterclass absolutely free and also share this link with your friends so that large number of people attend this masterclass and are benefited from it. Which of the following is insulin dependent? A GLUT1, B GLUT2, C GLUT3, D GLUT4. Only GLUT4 is insulin dependent. This GLUT4 is present on membranes of peripheral tissues, that is skeletal muscle, cardiac muscle, and adipose tissue. It helps in insulin-dependent glucose uptake after meals. So the correct answer here is D GLUT4. Cause of HHH syndrome. Hyperornithemia, hyperammonemia, homocystinemia is A. Deficiency of ornithine transcarbamylase, B. Defect in ornithine transporter, C. Deficiency of ornithine aminotransferase, D. All of them. Defect in ornithine transporter leads to HHH syndrome. This prevents ornithine transport into mitochondria and hence hits the cytoplasm and its reduced ability to clear carbamyl phosphate and enamel. So the correct answer here is B. Defect in ornithine transporter. Which method is used to separate a mixture of lipids? A. Isoelectric focusing, B. Page, C. Chromatography, D. Electrophoresis. It is Page. It is gas liquid chromatography. Wrinkled paper appearance of macrophage in bone marrow aspirate is seen in A. Tay-Sachs disease, B. Gaucher's disease, Niemann-Pick disease, leukodystrophy. It is seen in Gaucher's disease. In CRISPR-Cas9 system, which repair mechanism is used for genome editing? A. Homologous repair, non-homologous end repair, mismatch repair, nucleotide excision repair. It is non-homologous end repair. Rapport looping cycle shift oxygen dissociation curve to right because of A 23 DPG, 13 BPG, 3 phosphoglycerate, 1 6. It is 23 BPG. Hot flashes are experienced as a result of A. Increased noradrenaline, decreased estrogen, increased noradrenaline and decreased estrogen, increased noradrenaline and estrogen. It is due to increased noradrenaline, decreased estrogen. Restriction fragment length polymorphism is used for A. Analysis of DNA fragment, B. DNA estimation, C. Synthesis of nucleic acid, D. Detecting proteins in cell. RFLP or restriction fragment length polymorphism is used to identify a change in genetic sequence that occurs at a site where a restriction enzyme cuts. RFLPs can be used to trace inheritance pattern, identify specific mutation, and other for molecular genetic techniques. Thermogenic food is which of the following? A. High protein diet, B. High carbohydrate diet, C. High fat diet. It does not depend on the macronutrient. It is high protein diet. Ketone bodies are not utilized by A. Brain, B. RBC, C. Heart, D. Skeletal muscle. In RBC, there is no mitochondria and ketone body utilization pathway takes place in mitochondria. So RBC cannot utilize ketone body. So the correct answer here is B. RBC. Enzyme deficient in hyperammonemia type 1 is A. Ornithine transcarbamylase, B. Arginase, carbamyl phosphate synthase 1, arginosuccinate synthase. It is carbamyl phosphate synthase 1. Please click the link in the description box to join the ultimate Neet PG Breakthrough Blueprint Masterclass. It is a 2-day masterclass. Type of mutation seen in sickle cell anemia is A. Insertion, B. Deletion, C. Point mutation, frame shift mutation. It is a point mutation. Which replacement of amino acid does not alter its normal function? A. Glutamine replaced by arginine, glutamine replaced by alanine, glutamine replaced by glutamate, glutamine replaced by asparginase, glutamate replaced by asparginase. The clinical feature below occurs due to C. It is a Casal's necklace. The four options are tryptophan, phenylalanine, cysteine, glutamine. It is tryptophan. Which of the following binds to mRNA with ribosome in eukaryotes? A. Poly A tape, B. tRNA, S. methyl guanine cap, shine diagonal sequence. It is S. methylene guanosine cap. Which of the following is anaplerotic reaction? A. Conversion of pyruvate to lactic acid, conversion of pyruvate to oxalacetate, conversion of pyruvate to acetyl CoA. It is conversion of pyruvate to oxalacetate. Which of the following groups of protein is cysteine, folding of other proteins? A. Proteases. It is chaperones. Chaperones help in protein folding. Myoglobin has which structure? A. Primary, secondary, tertiary, primary and secondary, primary, secondary, tertiary, quaternary, primary, secondary. It is primary, secondary, tertiary. Best investigation for metabolic disorder is A. Western blot, B. Tandem mass spectrometry, PCR, gelatin. It is tandem mass. Separation of protein by their mass is A. Gel filtration chromatography, ion exchange chromatography, isoelectric focusing, ablative chromatography. It is gel filtration chromatography.